Welcome to topic 10 in marine and aquatic ecology. And this topic continues at the population level. And we're going to continue a bit with predator-prey interactions and unstable oscillations and outbreaks. And outbreaks of, for example, diseases, which is an isocline theory that is specific to epidemiology, very similar to the Lotka-Volterra equations, but we won't go into too much detail on that. And then we continue to unit 3 of the class, which is at the community level, in which we're going to look at some properties of communities related to energy flow. So the food chain length and trophic structure, food web structure in communities, and how to quantify energy flow through ecosystems. All right, our next topic within the population ecology unit and consumer resource interaction between two populations is the question of unstable oscillations between the predator and the prey population. It's possible for oscillations to be unstable, in which case they can increase in their magnitude. And these types of oscillations might be important in explaining extinctions in some predator-prey systems. Oscillations that are the opposite of damped, but rather increase in amplitude, with eventually one species eliminated. And the species usually eliminated in these wide oscillations is the prey species. That's because while the predator can usually switch to a different prey when one prey becomes rare, the prey can't switch to another species of predator when one of them becomes abundant. It's just at the mercy of whatever predator is there. The most vulnerable prey species to extinction by the predator is a species that shows positive density dependence when at low density, or a positive correlation between density and mean individual fitness at low density. So that would look like this. That would look like a prey isocline that is increasing, actually increasing as the prey abundance increases and the predator abundance increases. An increasing prey isocline like this. So in this case, if you start right here in the upper left quadrant, then we're going to go down a lower abundance in both species until we reach the prey isocline here. And then the prey is going to increase while the predator continues to decrease. Then both increase. And then above here, the predator continues to increase while the prey decreases. And look, we've got an unstable, increasing amplitude oscillation. So this is an example of what can happen if you've got self-acceleration in the prey. And what that means is when you hit the prey isocline on the up phase again, notice we're even higher in predator abundance than we were down here when we hit the prey isocline before. All right, so now we've got, when we come back around, both species are decreasing, we have the risk of hitting the y-axis here, and the prey has gone extinct. All right, so this could happen if you have this humped prey isocline here that in which you have to have some positive amount of prey to start out with before the prey population starts increasing here. So anywhere in this region, the prey population is going to be decreasing. So if there's any prey at this low abundance, it's just going to decrease down to zero. So you need to have some minimum amount of prey before the population can start to increase. So this is a positive correlation between density and mean individual fitness at low density. And this is also called undercrowding or depensation instead of compensation, and is also called the Ali effect, after Warder Ali, who published a paper in 1932 showing that goldfish grow faster when there are more goldfish in the tank, which was kind of unexpected. So you need to have some prey to begin with before the prey can start increasing. So in this case, there's some region of the prey isocline that is actually has a positive slope. A positive slope. So it would appear that there's a positive relationship between the prey fitness and the predator abundance. So the idea here is that the prey fitness actually increases. The fitness of the prey actually increases as the prey population density increases for this small region, or this limited region here, when the prey has a relatively low abundance. And then as that abundance increases, we get the usual logistic relationship with a decrease in fitness as prey increases in abundance. So this portion right here, this little portion right here, is the opposite of logistic growth, which assumes that mean fitness is highest at the population size of 0, which means that we should go up here, and declines along a straight line as population density increases. Instead, in the Ali effect, mean fitness also declines to low values when the population size decreases down to here. At low density, fitness is low. Then, as density increases, fitness increases to some maximum at intermediate density. Then, as density increases more, fitness eventually declines again to 0 at carrying capacity. So the plot of R against density is a humped plot, low at both ends, but higher in the middle. And in this case, the isocline is also a humped plot. The isocline may intersect the x-axis at 2 points, at the carrying capacity at high density, and a second unstable equilibrium at low density. So at low density, there's a density at which the population growth is 0. But if the population increases slightly, the growth increases. If the population decreases slightly, then the growth can only decrease until the population goes extinct. So that's the Ali effect. So if the population falls below some critical threshold, N, right here, it is doomed and eventually goes extinct. So Ali found this effect in goldfish in a tank exposed to levels of silver that are toxic to a single goldfish. However, if he added more goldfish to the tank, the same levels of silver were no longer toxic. So apparently, addition of more goldfish caused them to share the absorption of silver, so that each goldfish had absorbed less silver on average. Okay, so that's what he found in this experiment. But of course, there are many other mechanisms for an Ali effect other than what he found with silver. So for example, one is cooperation. So if individuals cooperate to increase fitness, then fitness might increase as more individuals are present and allow cooperation. If there are too few individuals, there's not much cooperation, so your fitness might drop to 0. Also mating. If the population is too sparse, then males and females might not be able to find each other to mate. So if the population goes too small, then it's doomed to extinction. Also chemical and nutrient flow. If the population actually fertilizes its food supply through death and decay of its own individuals, then low density can result in lower food supply and lower mean fitness. As, for example, Pacific salmon, who die at their spawning grounds after laying eggs, and whose juveniles hatch and feed on the invertebrates whose productivity may have been enhanced by the decaying bodies of their parents. So if the population gets too small, there's not enough fertilizer in the stream and not enough invertebrates, so those juveniles can't make it. So that's an example of an Ali effect. So in prey populations showing an Ali effect, if the predator drives their populations below the threshold density, this density right here, necessary to maintain the prey population, then the prey can go extinct. And this process might be responsible for many local extinctions and probably most complete extinctions, such as the passenger pigeon and the Steller sea cow, which were hunted at very small densities by humans. They drove the population down to very small densities that were too low to allow the population to recover. Once you get to a really low density, the population can no longer recover on its own. And so you have to have captive breeding programs to help it recover, which didn't exist at the time. So in these cases, the Ali effect mechanism was probably collapse of reproduction caused by collapse of the social system of these species, of the passenger pigeon and the Steller sea cow, and inability of individuals to successfully mate. So if you have the Ali effect here, this positive prey isochline that intersects the y-axis, comes down to 0 on the y-axis, then the result can be unstable oscillations and ultimate extinction of the prey. So here's an isocline crossing where there's no self-limitation, and we have increasing self-limitation as we change the angle here. So we have no oscillations here, complete self-limitation. So here we have undamped oscillations or permanent oscillations. And as you get more and more self-limitation, that damps out the oscillations and you get no oscillations at all. But then if you continue to rotate it, you get this situation where you get accelerating oscillations, self-acceleration in the prey, which caused wider and wider oscillations, unstable oscillations, which likely will eventually cause extinction in the prey as the system moves towards the y-axis and intersects the y-axis here, which means zero prey, an extinction of the prey. So with a humped prey isocline, we have self-accelerating prey population at low density, which results in unstable oscillations, or can result in unstable oscillations and extinction of the prey. But as we've seen, it's also possible and maybe even common for prey to have a refuge at low density, either a space where a few prey can escape from predators, or a refuge in numbers where the density is so low that it no longer is an advantage for the predator to target that prey, and they switch to another prey. And this allows them to escape from predatory extinction. A refuge at low density allows them to escape from the predator and protects them from extinction. But that refuge must exist at a population density high enough for the prey population to continue to maintain itself, to successfully find each other and reproduce and have a social system that allows the population to continue to increase. So that refuge has to be maybe at a low density, but still a high enough density to overcome the Ali effect. Okay, so that's probably more common. All right, our next topic in studying the consumer resource interaction, this is outbreaks. So an outbreak is a sudden explosion in the population of the consumer. which can be a predator, a grazer, a parasitoid, or a parasite, a disease. So these can be devastating to the prey populations and can cause extinctions in the presence of an Ali effect. So what is the cause of outbreaks, and can we understand them on the basis of the isocline geometry? All right, there are several possible causes of outbreaks. Number one, perhaps the most common, is simply environmental variation that occasionally, in space or time, increases the fitness of the predator, allowing the predator population to increase. In terms of the isoclines, this could simply result in an increase in the predator isocline, a vertical rise in the entire isocline in the phase space diagram. So here we have a phase space diagram for the two isoclines. So for example, you could have a predator isocline that looks like this. Um, it saturates fairly quickly, and the equilibrium here where the two lines cross is an equilibrium where the predator population is relatively small and the prey population is relatively large. So the population is going to oscillate or fluctuate around this equilibrium right here. And then suddenly the conditions improve for the predator. So its fitness increases, so its isocline rises way up like this. And now there's a new equilibrium here where the predator is much more common and the prey population has been knocked way down to low levels. So there's fluctuations around this equilibrium here, which could result, if fluctuations are wide enough, they could result in extinction of the prey population. All right, in the Mediterranean, probably a common example of this phenomenon is the development of sea urchin barrens in many areas. So urchin outbreaks cause widespread destruction of algal populations, with it converting lush macroalgae populations to bare rock, a rock covering of turf algae. So here's the urchins here, surrounded by an urchin barren. So the urchin outbreaks can be caused by external changes that favor the fitness of urchins regardless of prey density, such as local pollution that reduces the fitness of urchin predators. And so the predators become less common, so the mortality rate of the urchins goes way down, so the population explodes. Or there can be local overfishing that depletes the population of urchin predators, which also causes mortality rates of the urchin to go down. So this increases the entire urchin isocline, allowing it to increase to greater densities than others. So this can happen in some places but not others, or sometimes and not others, so that urchin barrens can be an indicator of either overfishing or a change in conditions that harm the biotic enemies, probably mostly fish species, the predators of urchins. Second cause of outbreaks, in many cases, if the prey isocline shows a humped shape and a substantial region over which the prey isocline has a positive slope, again, this is an Ali effect, a positive relationship between prey density and fitness, then that positive slope means that predator and prey can both increase at the same time. Prey populations increase because increasing density favors the prey itself, so more mating opportunities and cooperation increase fitness. And the increased prey then causes an increase in the predator population. All right, so this situation has been hypothesized to occur for the crown of thorns sea star and its predators, the large triton and the pufferfish. So you can read about in this paper by McCallum, which they show the isocline plot here. So this is the prey, and this is a predator. And in some cases, we've got the isoclines crossing in two places. So there are two equilibrium points. So this is the outbreak point, and then this is the low density point. All right, so we can draw our own isocline diagram here and make it even better. And can add more realistic details. Okay, so the prey itself increases over some positive region, like this right here, which causes the predator outbreak. So this occurs when the slopes of both the predator and the prey isoclines are positive over a large region of the prey density. So this geometry can create two possible outcomes. First, you got wide permanent oscillations in predator and prey that are regular and predictable. Second, we have sporadic outbreaks in the predator that are caused by small environmental changes. And this latter effect can be caused by the phase space diagram of the two isoclines cross each other in two places, one at high prey density and one at low prey density. So if they cross each other in two places, and that means there are two possible equilibrium points, and the population can cycle back and forth between these two equilibrium points here. In this case, we actually have three equilibrium points. And so this situation is hypothesized to explain the sporadic outbreaks in the crown of thorns sea star. And one of its predators. So it could be the giant triton, or the triggerfish, or pufferfish in the Indo-Pacific coral reefs. Okay, so we have outbreaks here in the prey species, which in this case is the crown of thorns, which is also a predator itself on smaller animals. All right, the third possible cause of outbreaks, we're talking about disease outbreaks now, is the loss of immunity in the host to a contagious disease vector, so similar to the Red Queen. So loss of immunity can occur in three ways. Sometimes it's a normal part of the immune phenomenon. So immunity can be temporary and gradually become weaker until the host now is susceptible to the disease again. So environmental conditions can change, such as warming or more acid ocean, and weaken immunity in the host, or bring a new pathogen into the environment to potential hosts who have never evolved with it. As for example, as the ocean warms, more tropical parasites can move poleward and initiate new outbreaks. Often as part of the Red Queen process, the pathogen mutates, and the mutation makes it once again invisible to the host's immune system, allowing it to infect the host again. So this, of course, occurs again and again, as we know from our own experience with the common cold and winter flu epidemics. We can become immune after being infected, but next winter, a new mutant form arrives that we're not immune to. Of course, the coronavirus, COVID-19, apparently is a mutant strain, a new mutant from a bat coronavirus. So once immunity is lost, the host population is susceptible and can be infected. And if the pathogen is contagious enough, it can sweep through the population in an epidemic. And then this is an outbreak of the disease, and it can be a pandemic. So epidemics of contagious disease occur often in the marine environment. The most well-known are probably the wasting diseases of sea stars and eelgrasses. So the eelgrass wasting disease that wiped out 90% of Zostra populations in the Atlantic Ocean in the 1930s, caused by a slime mold, Labyrinthula zostrae, and there are occasional outbreaks still. So there was one in the late 1980s that caused devastation of the eelgrass habitat in Great Bay, New Hampshire. So compromised tissue has a little bit of this brown decaying tissue here that's not photosynthesizing. Dead tissue looks like this. Uninfected looks like this. Infected like this. So here's an example. Here's an eelgrass bed in the bay. You can see here, and the beginnings of the wasting disease. And over time, gradually, a large portion of the bed is killed off in just basically 1 year. And by a couple of years, something like 90% of the bed is gone. So we know what the pathogen is. It's a slime mold. It's a marine slime mold. But we don't know why we have these recurrent outbreaks. It could be because environmental changes occur that make the eelgrass more susceptible to the pathogen, or it could be new mutations that the eelgrass is not able to overcome. The most recent sea star wasting disease outbreak in the Pacific began in 2013 and probably has run its course by now, caused probably by the single-stranded DNA virus, the sea star associated densovirus, SSA-DV. And again, we don't know why the outbreak happened. We know what caused it, the disease organism, but we don't know why. It happened just here and it not on either side of this peak, possibly because of change in environmental conditions that favored the pathogen or made the host more susceptible. And now we've got immunity has evolved, or there could have been a mutation that the hosts were not resistant to. So genetic possible or environmental cause of this outbreak. So there are many diseases targeting marine species, including commercial seafood species, aquaculture species, affected by bacteria, protists, viruses, and others. And this is a great review article here by Lafferty and Gerber, describing a lot of these. For example, Vibrio bacteria in many invertebrates and fish, dermo in oysters, worms and herpes in abalone, white spot disease in shrimp, betanoviruses in groupers, hematopoietic necrosis virus, Kudoa and sea lice in salmon, for example. So lots of potential for research on these dynamics in marine species. All right, the first mathematical treatment of infectious disease spread is as well known as the Lotka-Volterra competition and predation equations, and this is the Kermack-McCormick SIR models of infectious disease, which were first described in 1927 and are still used today with a few variations to understand the course of an epidemic, including the coronavirus. So these papers may have been inspired by the Lotka and Volterra papers published a few years earlier. And here's a recent review by Fred Bauer at the UBC of these models, discussing them. Good one to take a look at. So these SIR models, SIR models, are very simple, simpler to understand really than the LV models, Lotka-Volterra models. So the basic idea is that there are three types of hosts in the population, S, I, and R. S are susceptible individuals who have no immunity. I are infected individuals that are capable of passing the disease to S individuals. R are recovered individuals who had the disease and can no longer spread it because they're now immune. Well, they also could be dead. Either way, they can't spread it anymore. And the population starts out with 100% S susceptible when the disease arrives in one or a few individuals. And the disease arrives maybe as a new mutation in a coronavirus, for example, that nobody is resistant to, never seen before. So the number of susceptible individuals declines to an equilibrium that's set by the degree of contagiousness of the disease and the regular contact between infected and susceptible individuals. So the more contagious the disease, the higher the proportion of the population that eventually becomes infected. and the higher the eventual mortality. Eventually, a condition called herd immunity is reached. This is when the number of susceptible individuals drops to a threshold at which the disease can no longer spread, and then the epidemic is over. So the reason spread stops is because the rate of spread is proportional to the population density, and there's a threshold in the population of susceptible individuals. If the number of susceptibles is above the threshold, then the disease continues to spread. If it drops below the threshold because of mortality or immunity, then the disease stops spreading. So it basically kills out the susceptibles, and so it no longer has enough hosts to spread, or it converts the susceptibles to resistance. So it, again, no longer has enough hosts to spread. So the threshold number of density of susceptibles where herd immunity is reached depends on how contagious the disease is and how much contact is made between individuals that allows the disease to spread. So here we go, just a couple differential equations, and these define the isoclines. Plot the isoclines and watch the disease course through the population. So the threshold of herd immunity is defined by the basic reproductive number of the disease, R0, which is the number of individuals that one infected individual infects at the beginning of the epidemic. So the disease spreads if R0 is greater than 1 when all individuals are susceptible. So herd immunity is reached when the average infected individual infects just one other individual before dying or becoming immune. All right, so you can calculate the proportion of the population that needs to be infected in order for herd immunity to be released, and that's just 1 minus 1 over R0. So if each infected person infects 2 other susceptible people, then R0 is equal to 2, and the proportion of the population that needs to be infected to get to herd immunity is 1 minus 1 over 2, which is 1/2 of the population. If R0 is 3, one person infects 3 people, then 2/3 of the population needs to be infected to get to herd immunity. If one person infects 4 people, then 3/4 of the population needs to be infected to get to herd immunity. If one person infects 10 people, then 1 minus 1/10, or 90% of the population, needs to be infected before you get to herd immunity. Okay, we have looked at the competition interaction, the negative-negative, minus-minus interaction, and we looked at the consumer resource interaction, the plus-minus interaction, using the Lotka-Volterra models. And what we have left are these, the detritivore detritus and the mutualism interaction. We're not going to look at these in detail using isocline models. So this interaction is not really an interaction because one of the species is not affected by it. So this is just so that this species can actually be considered kind of like a resource in a logistic growth equation. So a single species logistic growth equation. So that's how this interaction can be treated mathematically. A mutualism is a true interaction, so both species affect each other positively, so both species can increase the other species' population, but obviously not without limit. Eventually, some other factor becomes limiting. Some other resource becomes limiting. So habitat, space, or food becomes limiting. Some factor outside the mutualism eventually stops the dynamic. Okay? And so we're not going to look at these in any more detail, but instead I have linked to some great videos you can watch to see examples of all of these interactions. So take a look at those. All right, so this wraps up the population unit of this course. We've run out of time talking about populations. So as we've seen, population ecology is mainly a mathematical study of the properties of populations. So population growth, population density, population dispersion, and how those properties affect fitness of organisms in the same population and in other populations. And those mathematical dynamics are described using differential equations that describe the rate of increase of population density due to density of the same species and the density of other species weighted by the effect of other species, whether that effect is positive, negative, or zero. All right. And studying populations helps us to understand basic adaptation and evolution of adaptations in any species. And it also helps us understand applied aspects of these concepts. So sustainable harvest, conservation, um, recovery of rare species, protection of vulnerable species, all involves some aspect of population ecology, understanding how populations increase and decrease in response to other species and in response to the environment. All right, now we move to the next level of organization in biology, from populations to communities. So we move from one subfield of ecology, population ecology, to another subfield of ecology at the community level. So here at the community level, ecologists use different methods and have a different focus, not on population level properties and phenomena, but with community level properties and phenomena. Then there are three major subfields of ecology at the community level. These are called ecosystem ecology, community ecology, and landscape ecology. Ecosystem ecology focuses on flow of nutrients and energy through a community. This is primarily a chemical subfield of ecology. Flow of nutrients and energy through a community, and creates mathematical models that describe that flow and allow predictions about all sorts of things in that ecosystem. Community ecology focuses on species, their taxonomic richness and functional richness and diversity, and creates predictive models about biodiversity. Landscape ecology is a sort of hybrid between ecology and sociology, sort of a multidisciplinary study of all ecological processes at the landscape scale, like over the entire Adriatic, but mainly focusing on biotic diversity, its economic value, and how to maintain and conserve it across geographical landscapes containing all kinds of biological, cultural, political, economic mosaics. So we won't be looking at landscape ecology, as that's a huge subject that's outside the scope of biology. So we're just within biology here and not considering sociology. In terms of Darwin's struggle for existence, the community level of focus is inherent in the idea that resources become depleted during the struggle for existence. That species become adapted to ecological conditions and resources over evolutionary time. And among the things they become adapted to are other species with which they interact. We can think of a community as the evolved result of the struggle for existence. The species in the community must be in some sense in harmony with each other, as they have evolved the ability to coexist with each other, to survive and thrive in this struggle. And therefore we can study species to understand how they have become adapted to the community in which they live. What's their place in the community in which they live, or how communities become assembled in nature in terms of the functional traits of the species that live in the community. Species has a role, a function in the community that may promote the coexistence of those species and the persistence of that community. So we can think of communities as assemblies of compatible competitors and predator-prey systems that have found sustainable ways to divide up resources at a particular location without depleting them or causing extinction of any member of the community. So at the community level, the subfield called ecosystem ecology is the field that specifically studies the cycling of nutrients and the flow of energy through the community of species at any particular location. All right, so first let's define community. A community is any local collection of populations of different species that interact in a way that affects their average fitness. All right, so this requires that we're looking at species populations collected together at the same place and time, because a key assumption is that they're capable of interaction. Of course, there could be interactions that are beyond local, especially in the marine environment, since organisms can migrate long distances, especially larval propagules. And therefore, species existing at a great distance from a local community can affect the individual's fitness in that community through the effects of the migrants that it sends. So often we're making some simplifying assumptions, and these can and have been criticized. And so communities can be defined by, you can define a community by all species within a location, a subset of the species within a particular location that interact in a particular way, and this is called a guild. There are all kinds of guilds. The dominant species within a location, and it can be defined according to the abiotic factors or conditions that are geographically correlated and are important in determining the type of species that exist in the community. So, for example, we can define the rocky intertidal community or the abyssopelagic community, the mangrove community, rocky intertidal community, the coral reef community, the kelp bed community, the salt marsh community, the salt marsh community underwater, or the intertidal mudflat community, intertidal sandflat community, the sandy beach macrofauna community, the myofaunal community, the community of organisms that are roughly the same size of sand grains on a sandy beach that you can see through a microscope, the myofauna. Myo means sand, sand fauna community. Abyssopelagic community. So if we say the sandy beach macrofauna community, that means here we're interested only in the interactions among the macrofauna, assuming it's reasonable to restrict our analysis to just the macrofauna. It may not be, but if you make that restriction, that means that's what you're interested in and that's what you're studying. So obviously, if we just want to describe the species that are present, then there's nothing illogical about that. But if we want to understand all the interactions affecting fitness, we will have to look at other species that share the habitat, including, all the groups. So if we're talking about the sandy beach, we're also going to have to include the myofauna because many of the macrofauna eat the myofauna, for example. So the basic goal of community ecology is to understand communities as a collection of complementary evolved adaptations. Within all communities, organisms have to survive and reproduce successfully within that community. And the survival and reproduction of organisms within the community inevitably creates properties that we can observe at the community level. These properties at the community level can then feed back to organisms and affect their fitness. And the cycle continues. Each level of organization, organisms, populations, and communities has emergent properties, as we've talked about, at that level that are not observable at the lower level. So at the community level, several properties that we will consider are these: trophic structure, productivity, biomass, efficiency, trophic efficiency, species diversity and richness, community similarity. All these community level properties are evolved properties that are consequences of the adaptive evolution of species within the community. Adaptation at the level of populations will inevitably affect these community level properties. And we can understand communities as evolved collections of adaptive functions in the species that compose the community. So we started this class with the concept of fitness and abiotic and biotic effects on fitness. So abiotic modifiers of fitness include conditions such as light, temperature, and salinity. Biotic modifiers of fitness include within and between species modifiers. So within species, an individual's mate, its family members, and non-relatives within the population affect its fitness. Between species, competitors, predators, mutualists interact with members of that species to influence its fitness. And if we look at the community level, we immediately see that there's an additional distinction between types of modifiers on fitness. We have direct and we have indirect effects. Indirect effects of other species on an organism's fitness might be more common than direct effects. Acknowledging that species exist within the context of a community immediately shows us that species are part of a wide network of indirect effects, a network of cause and effect that requires a new kind of analysis. And the community property we will start discussing is trophic structure, which is the overall pattern of feeding relationships within a community. So let's demonstrate that we cannot understand the effect of species 1 on the fitness of species 2 without an understanding of the trophic structure of the community, and therefore the potential indirect effects. So let's say we have a predator and prey. We can draw these as compartments symbolized by a circle with predators above the prey, and we connect these two compartments with an arrow pointing from the prey to the predator, indicating that the predator consumes the prey. And the arrow indicates the direction of transport of energy and materials from prey to predator. So in community ecology, we're modeling individuals as compartments in a system of flow of energy and materials. So we've got this compartment right here, this compartment right here, and they're connected by a directional flow, a chemical flow of energy and materials. We also follow the convention of drawing the predator above the prey, flow of energy and materials is always drawn in the upward direction in food web diagrams. So here's a simple trophic structure. A single top predator species, red circle, that feeds on two species, orange circles, who themselves are both competitors feeding on a single resource population in the green circle. Green to indicate plant, therefore two competitors here are grazers. So here's a question. Assume the grazer species 2 is a superior competitor over species 1. If it is allowed to reach its shutout level, it will competitively exclude grazer species 1. But it's prevented from reaching its shutout level by the predator because the predator prefers species 2, so it keeps its numbers down so that it doesn't reach the shutout level and shut out species 1. So here's the question. What effect does this predator have on grazer species 1's fitness? Remember, it is a predator of species 1, plus it also is a predator of species 1's enemy, the superior competitor species 2. All right, so now you can see how indirect effects can greatly complicate fitness relationships. If you take into account the community context, you can see those indirect fitness effects. In this case, the effect of a predator on its prey, grazer number 1, is not really clear. It could be positive or negative. Overall, obviously predation is a gross negative effect on the prey, the direct effect, but preying on its superior competitor is a gross positive effect. What is the overall net effect of the predator on grazer 1? Well, we don't know. It could be positive or negative. The predator could have a net positive effect by consuming its competitor and thereby allowing grazer 1 to continue to exist where it would otherwise be excluded. Or it could have a net negative effect by virtue of its basically consuming and killing grazer 1. So which effect is it, positive or negative? Well, to answer this question, we'd have to analyze the 3 differential equations describing population growth of the 3 species, assuming we have enough data to understand the parameters of the equations and the magnitude of all 6 direct effects on fitness. Or we could do a field experiment and remove the predator at some locations and monitor the effect removal had on grazer 1 compared to non-removal control locations. And this particular trophic structure is very, very realistic and occurs commonly, as for example, baleen whales that feed on both copepod and mysid crustaceans, which themselves compete for phytoplankton food. So it's a very simple structure, and even for this very simple structure, the fitness relationships might be difficult to determine because of indirect interactions. Here's another example, another very simple trophic structure. Again, even though it's very simple, it's hard to say whether the effects are positive or negative. So we have a simple 3-species community. A top predator, species 1, red circle, feeds on a prey species 2, red circle, which is also a predator that feeds on a resource population 3. But the top predator 1 also feeds on the bottom prey 3, in addition to feeding on the predator 2 of prey 3. So what effect does predator 1 have on the fitness of prey 3? The direct effect is negative. Predator 1 feeds on prey 3, so obviously it's going to reduce its fitness by killing the prey. But it has a positive indirect effect on 3 by feeding also on 3's enemy, namely predator 2. What's the net effect of predator 1 on prey 3's fitness then? It's the sum of that negative direct effect, the positive indirect effect. And we don't know what that sum is unless we have more information, enough information to model the dynamics of all three populations, or information from a field removal experiment. If we found that removal of predator 1 causes an increase in population of prey 3 relative to controls, then we might be able to conclude the net effect was positive. So again, this trophic structure is extremely common in nature. For example, salmon feed on smaller bait fish such as sardines, herring, anchovies, smelt, but they also feed on the prey of these bait fish, such as krill. So what is the net effect of salmon on the krill population? Well, it could be very well be positive overall, even though it directly kills the krill by preying on it. Okay, so we have the concept now of the beneficial predator. Many examples of this. Chitons, for example, feed on seaweeds. Sea stars feed on mussels and barnacles. And indirect interactions can be a strong determinant of an organism's fitness. The chitons feed on seaweeds that are dominant competitors, but they also feed on seaweeds that are weaker competitors that benefit from their feeding on the dominant competitors. Same with sea stars and mussels and barnacles. In feeding on the mussels, which are the dominant competitors, sea stars may maintain the populations of barnacles, even though they feed on the barnacles. All right, so even simple trophic structures can result in indirect interactions that start to become complicated. So consider the consequences. So consider the consequences when you actually look at a real community in nature, rather than these simple models where the possible indirect interactions now become basically infinite. So here's a real food web. Are you ready for this? There it is. This is a cod-based food web in the North Atlantic. So this figure here shows a tangled mess of feeding relationships with hundreds of predator-prey connections. If a simple 3 to 4 species trophic structure is difficult to understand, then how will we ever be able to possibly understand such a complex food web? So here's another one. This was done within our program that was from a package that's designed to analyze food webs. All right, well, that's a good question. How can we possibly understand this? Well, ecologists have been grappling with that question for over a century. And this question is basically at the heart of an ongoing debate in ecology over the relative merits of a holistic versus reductionistic approach to understanding nature. The basic question is, can organisms and their adaptations be understood in isolation, or can we not understand them without taking the entire community into context? So often it certainly seems that you can't really make predictions without taking the entire community into account, which usually means you can't make accurate predictions at all. And so there's a debate between holism and reductionism in ecology. You can't understand one interaction without understanding all interactions, which basically paralyzes you, your ability to understand anything. So as Garrett Hardin said, you can't do just one thing. If you try to understand one component of the system, you find out that you need to understand another component before you understand that component, and so on until you end up needing to understand the entire community. And we have the law of unintended consequences. This is the idea that you make a small change in the community, and that leads to many unanticipated consequences because you don't anticipate them because you don't understand all of the indirect effects, which can be unlimited number of indirect effects, which are very difficult to forecast and predict. So consider, for example, the case study of the sea walnut. This is a ctenophore, Nemiopsis leidii, and its introduction into the Black Sea community in 1988. So this species is a comb jelly, phylum Ctenophora, native to the western Atlantic Ocean, a predator feeding on small zooplankton, protists, and mesozooplankton that it captures with its tentacles. But the diet also includes ichthyoplankton. This means the larvae of fish that live in the plankton. So the larvae of anchovy, sardine, herring, these fish species also feed on plankton. This jelly has predators in its natural range in the western Atlantic, including several fish species like the southwest Atlantic butterfish, Stromatius brasiliensis, and the choicey ruff, Serialella porosa. And these predators apparently regulate its population and maintain it at relatively low levels. In the early 1980s, this jelly was introduced from its native Atlantic into the Black and Azov seas from ballast water that was loaded in shipping vessels in the Atlantic and then discharged at destinations in the Black Sea. So discharge of ballast water in this way is highly regulated now, much more so than in the past, because it has the capability of introducing huge numbers of non-native species that have a life stage in the plankton capable of surviving that transport in the ship. And many jellies have been introduced in this manner in many areas because they often are selected species with a high growth rate and high fecundity. And they have a broad diet, which enables them to survive harsh conditions in the ballast water for a long period of time. So Nemiopsis invaded the Black Sea in ballast water in the early 80s. And the Eastern Mediterranean, the Aegean Sea in the early 90s, and the Caspian Sea by 1999. So what impact on the Black Sea community would we have predicted the sea nut walnut to have? If we try to predict its effect based on its preferred diet, we might predict that it would probably cause a reduction in native small protists and zooplankton in the Black Sea, because that's what it feeds on, and perhaps compete to some extent with other jellies native to the Black Sea, and might threaten some of them if it turns out to be a superior competitor, just by looking at this simple trophic structure here. So the jellies feed on the phytoplankton and the zooplankton, and the fish also feed on the zooplankton. And these small fish also are food for larger fish. And everything eventually ends up being consumed by the reducing bacteria after it dies and becomes detritus, which then produces nutrients that support the phytoplankton, which then supports the zooplankton. So here we have a nice simple community, nice simple trophic structure. So if we were to make a prediction based on this, we might say, okay, well, it's going to reduce the phytoplankton and zooplankton in the Black Sea community, and maybe compete against other jellies that are trying to do the same thing and might cause a reduction in the population of native jellies. So we have an introduced exotic that might compete against natives and cause a decimation of the native populations. That might be our prediction, okay? Well, we would be wrong, because what actually happened was introduction of this jelly into the Black Sea caused a collapse, almost complete collapse of the Black Sea anchovy population. So after we understand this collapse, we can certainly look at the main properties of the Black Sea trophic structure and understand why this happened in retrospect. But hindsight is 2020. It was not predicted at the time. So here's the key fact here that was ignored. If you just consider the trophic structure, the sea walnut was both a competitor and a predator on anchovy. So the two were competitors because they both feed on zooplankton. The anchovy feeds on zooplankton and the jellies feed on zooplankton. But the anchovy eggs hatch into pelagic larvae. So the anchovy larvae is pelagic. It's in the water and it's in the zooplankton, in the water column. And the sea nut, walnut, and all the jellies feed on the zooplankton too. So they're also feeding on the larval anchovy. So they're both competitors with the anchovy, and they're also predators on the anchovy as a larva. So they're double enemies of the anchovy. So this trophic structure is similar to the second simple example we looked at just before, with the top predator feeding on the middle predator and also feeding on the prey of that predator. And so the top predator is a double enemy of species 2 in the middle, having both a direct predation effect and an indirect competition effect on it, both of which are negative. So the sea walnut has two negative effects on the anchovy through consuming its larvae in the zooplankton and also consuming zooplankton, which is a limiting resource of the anchovy. So clearly the sea walnut is a double enemy of the anchovy, and in retrospect, it's no surprise really that its introduction led to the collapse of the anchovy fishery. But still, you might think that's hard to predict even with that knowledge, because it's not just the sea walnut is a double enemy, but all the jellies are a double enemy because they all feed on zooplankton. So why would introducing just the sea walnut into the ecosystem have this catastrophic effect when the other jellies did not have the same catastrophic effect? All right, well, in reality, the story is not quite this simple. There were three other elements that also contributed to the anchovy collapse, and those were, number one, organic pollution of the Black Sea that was a major problem in the 1980s. It's been cleaned up since then. And two, overfishing of the anchovy population also was a contributing factor. And three, the absence of any predators of the sea walnut in the Black Sea. The first two factors apparently weren't sufficient to cause a collapse, though. Introduction of the sea walnut seems to be the straw that broke the camel's back. The pollution increased the carrying capacity of the sea walnut and allowed it to increase to very high density by basically fertilizing the phytoplankton. So the pollution is detritus, basically organic material, that is recycled by the bacteria into nutrients that cause a phytoplankton bloom. So that's called eutrophication. So the phytoplankton increased to a very high density by basically fertilizing the phytoplankton with nutrients and increasing food at the base of the food web, including its zooplankton prey. So pollution also caused a depletion of oxygen, but because jellies have no ability or need to move quickly, the lower oxygen did not cause any loss of fitness, whereas it did cause a loss in the anchovy. All right, so here's the anchovy here back in the halcyon days before the 80s. Just a few Aurelia jellies in the Black Sea ecosystem. And then suddenly we have this explosion of the sea walnut here. And coincident with the sea walnut, the anchovy basically plunges. Abundance plunges. Sea walnut is reduced. The anchovy goes up again. Then we have another burst of the sea walnut and reduction in the anchovy. So we probably could have predicted this outcome if we were able to write down and solve all of the differential equations involved for the sea walnut, anchovy, zooplankton prey, with all accurate parameters, including fishing mortality rate and increased carrying capacity of the sea walnut, and if we knew there were no predators. But of course, we did not know a lot of this information, which points to the fact that real communities in nature are enormously complex. And we can't begin to know everything we need to know to make absolutely accurate predictions with no uncertainty about the consequences of species addition or loss. And in some cases, the entire scientific community is surprised by the outcome. So, this is just one example out of many of our lack of understanding of the net effect of species on other individuals in the community. And there are many other examples. For example, predictions that the introduced invasive alga Caulerpa in the Mediterranean would cause catastrophic losses of native algal species. That did not happen. And we're still trying to understand why that didn't happen and that the invasion hasn't really caused much damage. Two, predictions that the introduced aggressive European green crab in the Pacific Northwest estuaries would cause predator losses of native species, similar to the impact of the sea walnut. And that, again, never happened. And the crab has basically died out there, just hanging around at very low densities. Why didn't it happen? Three, predictions that an introduced gill parasite of the blue mud shrimp in Pacific Northwest estuaries would have little effect on the population and would stabilize at low densities, when in fact it decimated the shrimp populations. There have been others that did not do that, and this one happened to do that. And again, nobody predicted that, and we still don't quite understand why. All right, recent reviews of the ecological impacts of invasive species, such as this nice one by Jennifer Molnar, have found that anywhere from 50% to 75% of introduced species have been harmful in causing loss or extinctions of native species, and that's through aggressive predation or competition. Most of these harmful introductions have been due to shipping and aquaculture. And the most harmed regions are the Mediterranean, the North Sea, Pacific coast of North America, and the Hawaiian Islands. But very rarely, if at all, is it possible to predict which of these 50 to 74% are harmful and which are completely innocuous. And so laws are designed to follow the precautionary principle and assume that all introductions are harmful and should be avoided. So the reason these impacts are difficult to predict, of course, is that communities are basically infinitely complex without limit, and large numbers of indirect interactions are there that have unknown strength. So this complexity of real communities in nature has triggered a controversy. Controversy has always been there, really, and it continues now between two views, basically holistic and reductionistic approach to understanding ecological interactions. So the holistic approach basically holds, people think that you can't really understand one interaction without taking into account all interactions. So as Garrett Hardin wrote, you can't do just one thing. Any ecological change will have consequences. There will always be trade-offs because there's a balance of nature. And any alteration of that balance will result in a new and different balance across the entire system, which is difficult or impossible to predict. Whereas the reductionistic approach, which is often the only approach that's possible, really, you can't really understand everything all at once. And that's the idea that usually a few interactions are dominant. And those determine a species' fitness and its variation in time and space, even though there may be an unlimited number of interactions in total. And so if we focus on those important interactions, that usually that will provide information that's useful for both a basic and a practical or conservation or economic understanding of the system. Okay, so this is how communities are studied, usually sort of in a reductionistic sort of way, because that's the only way it can be done. People make an attempt at holistic analyses, but they usually tend to be more descriptive rather than try to understand mechanisms. All right, so how are communities usually studied here, in 2021? Well, there's field surveys of patterns of diversity, biodiversity in space and time, how they change over time, and in different areas, different types of habitats. And these are basically descriptive statistical analyses. And most of these are considered to be holistic studies because you're looking at everything, at least everything of what you can see, not necessarily looking at mechanisms though. And there are field experiments in which populations are manipulated, usually removal or caging, followed by monitoring the response of other members of the community. There are computer models based on differential equations, but instead of a few populations, or two like we did in the population unit of the course, a large number, a realistic number, a realistic number of interacting equations can be analyzed, possibly now with high computing power. And at the community level, there's a subfield of ecology of the discipline called ecosystem ecology. And this is a technical term, ecosystem. And ecosystem ecology, the subfield is basically going out in the field and using the computer to study the flow of energy and materials, nutrients, elements through the community. So systems, the word system is a technical term, technical jargon. Systems biology is a type of study consisting of looking at compartments through which energy and materials flow, and the study of the dynamic flow and equilibrium properties of those systems, and studying how you can modify those systems in a way to preserve flows, which can be very necessary for the community to continue to exist, and also are necessary for human beings as and their dependency on the environment, to their dependency on the environment and dependency on those flows through their environment. So now we're going to take a look at ecosystem ecology. That's going to be our first major focus here at the community level. And this is the portion of the struggle for existence framework in which the resources are depleted as they move through members of the community. We develop mathematical models of the flow of these resources, which are consumed and altered by community members as they carry out their life processes. So ecosystem ecology divides the community into compartments or components, each of which processes energy and nutrients in a different and specific way that we can quantify with numbers. And these compartments are linked by quantifiable flows or fluxes of energy and materials. So compartments can be hierarchical. In other words, smaller compartments can be contained within larger compartments, and flows through the smaller components can be summed to get overall flows through the larger inclusive compartment. And the major assumption of ecosystem ecology and this systems analysis is the mass balance assumption. The mass never changes. The total mass remains constant. So it's a conservation of mass argument that allows us to quantify the flow of materials through the compartment. Conservation of mass, conservation of energy. The major compartment in energy material flow ecosystem models are inorganic versus organic. So inorganic can contain component compartments for each of the major elements, such as nitrogen, phosphorus, potassium, iron, silicon, in this component right here. And the organic compartment contains the major functional compartments for trophic flow. So that's the living compartment of the environment, of the ecosystem. So we've got consumers, producers, detritivores, detritus. So those are the living and dead components of the organic compartment. Detritus is dead organic material, and detritivores are those who eat it, so scavengers, bacteria, fungi, and these constitute the detrital food chain. Consumers are, as we've seen, predators, grazers, parasites, parasitoids, and producers are species that convert chemical or light energy into organic molecules. And for the most part, these are green species who photosynthesize. And these constitute the grazing food chain. So we have the detrital food chain, the grazing food chain. So let's consider first the grazing food chain, which is usually the best known and the best documented in most ecosystems. That's this food chain right here. Okay, so I'm using the term ecosystem here as we're discussing the subfield ecosystem ecology. For the purposes of this discussion, ecosystem simply means a system of compartments through which energy and materials flow, each compartment being a subset of a local ecological community and its abiotic environment. So the grazing food chain starts with the primary producers, symbolized by a green circle. Right here. Because most primary producers are photosynthetic, and photosynthesis uses chlorophyll molecules, which are a pigment and reflect in the green portion of the light spectrum. So primary producers are autotrophs, that means self-nourisher, because they create their own chemical energy rather than consuming it from other organisms. They can create their own organic chemical energy either from light, which is photoautotrophy, or from inorganic chemical bonds, which is chemoautotrophy. Primary producers are in turn the food source for the heterotrophs, who obtain their energy from consuming other organic tissues. Heterotroph means nourished from others. So if the trophic structure is a simple linear chain, extending upwards from autotrophs to successively higher heterotrophs, then it's a food chain, and we can easily define the levels of the food chain. So those grazing the autotrophs are first order consumers. These ones right here, they're grazing on the autotrophs. And they in turn are consumed by second order consumers, which in turn are consumed by third order consumers, and so on up the chain. The consumer at the top of the food chain is called the top predator or top carnivore or apex predator. That's the one at the very top of the food chain. Okay, so simple food chain. The number of trophic levels, starting with the autotrophs as number one, is called the food chain length. If the top predator is a third order consumer, then there are 3 levels of heterotrophs and 1 level of autotrophs, equaling a total of 4 trophic levels. Okay, 1, 2, 3, 4. And the top is a third order consumer because the consumers are here. 1, 2, 3, third order consumer. 4 trophic levels total, including the primary producers. Some examples of food chains and their compartments in the marine environment. So baleen whales are often just second order consumers, filtering krill out of the plankton as food, which themselves feed on diatoms in the autotrophic compartment. So just 3 trophic levels from single-celled diatoms to the largest animal ever to have lived, the blue whale. So upwelling regions are the only regions on Earth where marine mammals or commercial fish, right here, are at level 3 of the food chain. Usually many more levels are required before the prey are big enough to be successfully captured and support fish or mammal populations. And the reason for this is that the autotroph compartment consists of diatoms, chain-forming diatoms in upwelling zones. Diatoms shorten the food chain because their cells form actual chains that are big enough for planktivorous fish or krill to directly feed on. So these are then the food for commercial fish, salmon, or marine mammals, pinnipeds or toothed whales in the case of planktivorous fish, and baleen whales in the case of the krill. So the major planktivorous fish are herring, anchovy, sardines, whose jaws can gape widely to capture diatoms during the blooms in spring and summer. On the other hand, open ocean food chains have to reach 6 levels, roughly, before they can support commercial nectons, such as tuna and squid. That's because the zooplankton are quite small and don't become large enough. Megazooplankton to feed the fish until we've gotten to 4 trophic levels, roughly. Continental shelf food webs outside upwelling zones generally are 4 levels to the larger fish, so salmon and shark. Though in some cases, there are 3 levels to cod, since cod can feed on benthic suspension-feeding herbivores, clams and mussels. So first question, what determines the number of trophic levels in a food chain? Here we're talking about the total number of trophic levels, not just the lowest number where commercial seafood appears. So here's a question that highlights the feedback between fitness and community properties. In surviving, reproducing, and adapting, populations that compose the community collectively determine the properties of the community. Conversely, community properties themselves can be considered habitat features for all of the populations that live in the community. So, food chain length, or the number of trophic levels, is a property of the community, not of any organism or any population. Food chain length, though, arises from the properties of all organisms in the community, which together create the food chain with its link. And the length of the food chain, conversely, creates or removes trophic opportunity for species in the community. And we answer this question by considering a compartmental model of the food chain of a coastal upwelling area. Okay, and as an aside, I'm here using the term food chain, but in reality, very seldom is any trophic structure a literal chain. We're talking about the number of trophic levels in the entire trophic web, which often is drawn diagrammatically as a simple chain, even though in reality there are many more connections that give it a web-like structure, as we've seen. And it is possible to determine using chemical isotopes of nitrogen or carbon what the trophic level of any particular organism is in the food chain, and often it is not an integer, but it can be fractional. So a trophic compartment diagram is usually pictured with compartments, okay, obviously, whose shape reflects the trophic function of the compartment. So this is an example, Chesapeake Bay estuary. So they're hexagons for heterotrophs, and they're D-shaped compartments here for autotrophs, and a circle for outside exogenous inputs. Each compartment has an inflow path and outflow paths with the assumption of mass and energy balance. The total inflow is exactly equal to the total outflow. So this mass energy balance allows the construction of a very complex model of the ecosystem that's easy to describe and solve. So far easier than accounting for all individuals in a population with differential equations. So, in contrast to the basic methods and goals of population ecology, which is to track the numbers of individuals, in these diagrams, we're not interested in individuals. We're tracking the flow of energy and materials into and out of the compartments that are defined functionally and can represent a single population or a group of populations like oysters that are similar in trophic function. So it's a fundamentally different way of looking at the interaction between organism and environment, tracking flow of resources rather than population densities. The flow rates in these diagrams can use several different units, all of which are more or less equivalent. Okay, so here's a compartment diagram for a consumer. And we have flows of energy into the consumer compartment and flows of energy out. And so the total inflow is equal to the sum of all of the outflows. All right, the units all use some estimate of energy flow on an areal basis per year, usually meters squared per year. So the unit of energy flow is usually grams of carbon, grams of dry weight, kilocalories, or joules, but these are usually interchangeable with rough equivalency. So all of these are basically the same thing, just interconvertible using conversion factors. So 1 gram of carbon is equal to 10 grams of dry weight, roughly, or 5 kilocalories, or 21,000 joules. And those are basically commonly used conversion factors. They might be a little bit different in different situations. These are sort of ballpark numbers. So the first two conversion factors will vary depending on the species and tissues involved in the flux, fat versus carbohydrate versus protein, etc. But these we've got here we'll be using for purposes of this class. Let's start with the autotroph compartment. We'll assume the autotroph is a photosynthetic organism using chlorophyll to capture light energy. So we'll make it green. It's got some diatoms here. The energy input into the autotroph compartment is solar energy. And it's important to realize that the vast majority of light cannot be captured. Just a very small proportion is captured and used and converted into carbon during the process of photosynthesis. So let's say our autotroph compartment has a gross photosynthesis rate of this amount right here of carbon, grams of carbon per meter squared per year. So this is the amount of carbon that is fixed during photosynthesis. The light energy coming in was captured and incorporated into the chemical bonds of these organic or carbon-based molecules, mostly sugars, that are the products of photosynthesis. So roughly a few percent, maybe 2% of the total solar energy coming in is actually captured and fixed as carbon bonds in plants. The rest of the light eventually dissipates as heat down here and radiates back out into space, or is captured in the carbon dioxide in the atmosphere and contributes to global warming. So the input pathway in our example is 557 grams of carbon per meter squared per year, and this is the gross photosynthesis pathway. And that 557 ultimately becomes outflows through the autotroph compartment. So one outflow is respiration. In other words, the energy fixed in sugars may be used by the plant. That energy may be used by the plant for whatever functions require energy. So the building of defense compounds, the pumping of solutes, growth and differentiation, reproduction, etc. Once those sugars are broken down and the energy used, that energy is lost ultimately as heat. In our example, that loss due to respiration comes out to about 117 grams of carbon per meter squared per year, or you could express it in kilocalories or joules or dry weight. So the remainder of that 557 is the net production. So 557 minus 117 is 440 grams of carbon per meter squared per year. That's the net production after respiration. Respiration uses up some of those carbon bonds, and the ones that are not used up, those are the ones that are still there. And so that's the net production of the plant, the growth of the plant, or the reproduction of the plant. It's a plant material that has been produced during the year and remains at the end of the year. So it amounts to biomass. It's still sitting there, green biomass in the community. That's 440 in this example. I should note that these are just examples. The numbers could be quite different, obviously, in different communities. The next trophic compartment is the first order consumers who directly consume those members of the autotroph compartment. The total production they have available to consume is exactly that 440 that the autotrophs produced, the net production. That's the total available, but they don't necessarily consume all of it, of course. So we can say the outflow before we enter the consumer compartment is the not ingested outflow, which in our example turns out to be 352 grams of carbon. This leaves 88 grams of carbon per meter squared per year that is consumed by first order consumers. So of this 440 produced by the plants, 88 is consumed by these grazers. Grazers are first order consumers. So this is called ingestion, input. Consumers ingest this amount of carbon or energy. In consumers, there are two loss outflows, one more than in the autotroph compartment. One we've seen before, that's respiration. And the other is the fecal energy flow, called egestion, in contrast to ingestion. And the assimilation is just the amount of energy that is incorporated into the body of the consumer and is equal to the ingestion minus the egestion, or the fecal energy flow. And so, however, it can be broken down for energy. Any kind of function of the organism, the consumer that requires energy, needs to break down that assimilated carbon during respiration, which captures that energy and uses it. So a subset of that assimilated energy, or carbon, is actually used and eventually is lost as heat in the process of respiration. So the total ingestion minus egestion and minus respiration, that leaves the net production, or biomass left over, of the consumer's compartment. That's the total biomass that the compartment produces that year per meter squared. That biomass constitutes growth and reproduction and is one of several possible measures of the mean fitness of individuals in that compartment. Okay, so you can sort of think of mean fitness as, or production as, a proxy for mean fitness. And we can continue moving up the trophic levels of the food chain to the second order consumers and quantify all the same pathways as in the first order consumers. And in our example, the productivity of second order consumers, the energy left over and used for growth and reproduction, is only 6 grams of carbon per meter squared per year. This amount of productivity is not very large compared to the input at the bottom of the food chain. It's not very much for a predator to live on. This shows the connection between a community property and the fitness of organisms. A predator is unlikely to evolve to feed on prey on these to prey on these second order consumers, given how low their productivity is, unless that predator is highly mobile and can forage over a very large area. So I mean, it will consume these second order consumers, but most likely it's not the only thing that they can consume. They have to be flexible and also go for the grazers. So imagine you could only feed on that second order consumer trophic level, which is producing 6 grams of carbon per meter squared per year. So that's the only thing you could feed on. So what does that amount to? That's about 2 carrots per meter squared per year, 25 radishes, 5 bites of an apple, 5 sugar cubes, 15 peanuts, 5 mouthfuls of beer per meter squared per year. That's not much to live on. You'd have to cover a large area in order to live on that for 1 year. This illustrates an important principle, that is, the higher up on the food chain that you consume, the larger area that you must range over, and therefore the more mobile you need to be. So usually that means large mammals or birds. So this is one of the most important laws of energy transfer up trophic levels, and that's the loss of productivity with every trophic transfer. Net productivity at the autotroph level was 440 grams. Net productivity at the primary consumer level was 75 grams. And at the secondary consumer level was 6 grams. So note that only a small proportion of the productivity at one level survives to the next. That's because most of it, 90% of it, is lost due to respiration, number one, egestion loss, number two, fecal energy flow. So this law is basically called the energy pyramid law in ecology. The energy or biomass production per year passing up trophic levels gets smaller each level, and that amount is analogous to the width of a pyramid, which gets smaller as you go up the pyramid. So the losses are those energy components that are not consumed, not digested, but are respired. So that energy is actually used and eventually gets lost as heat. It's like 90% of the productivity. So note that this law applies to net productivity, or net production, not standing biomass. If all that productivity gets consumed, then the standing biomass at any one time for any level is going to be small. So while net productivity is a pyramid, standing biomass can be anything. Okay, so this is a pyramid of net productivity. Again, the pyramid of net productivity. Phytoplankton, krill, baleen whale. Here we go. Pyramid of productivity. Okay, but this is not a pyramid of productivity because it's not a pyramid, so it can't possibly be productivity. That would be violating the law, second law of thermodynamics. So what is this then? Well, this is a biomass diagram. So in marine systems, standing biomass is often an inverted pyramid with the lowest standing biomass at the bottom, the phytoplankton, whose production is very rapidly consumed, leaving very little biomass at any one time. So this loss here, like roughly 90% loss, is called Kozlovsky's law. She proposed in 1968. As a rule of thumb, the production of one trophic level is approximately 10%, only 10% of the production of the previous trophic level. It's Kozlovsky's law. That's the trophic pyramid, the production pyramid. So that's just a rule of thumb, the 10% rule. The actual number, though, is not necessarily 10%, and it's been measured in many communities, and it's anywhere from, like, say, 5 to 20%. So 10% a good rule of thumb. So if you look at the number of studies that have been done, the trophic level transfer efficiency tends to be around 10% on average, but there are examples that are lower than that, just 2%, and other examples that are higher than that, around 24%, but these are not quite as common as communities around the 10% level. All right, the implications of Kozlovsky's law for consumers: in order to maintain the same productivity, a second-order consumer must forage across 10 times the area as a herbivore. And a third-order consumer must forage across 100 times the area as a herbivore. And a fourth-order consumer, if there is one, must forage across 1,000 times the area that a herbivore forages over, if it's just focusing on the third-order consumers as prey. And there are practical implications for human food production of Kozlowski's law. The amount of land that must be cultivated to produce a given energy demand for meat is a minimum of about 10 times the amount needed from plant crops. So you do this nice little thought experiment and imagine Japan is an island. So imagine that all of the food the Japanese eat had to be made on that island and couldn't come from the outside. Well, if you do go through the calculations, you'd have to cultivate the entire island if you're only producing meat on that island for consumption. Whereas if the only food is cultivated crops, then you would cultivate a much smaller amount, maybe 1/10 or more of the country must be cultivated. So it's a big, big difference, and it's a very large effect that needs to be taken into account. All right, so Kozlowski's law is an illustration how the property of a community can constrain the evolution of consumers. So a low ecological efficiency means how much productivity is retained as energy moves up the food chain. So low efficiency prevents the evolution of predators occupying high trophic levels, unless they are very mobile and able to forage over very large areas. So this brings us to an important property of food webs, namely their ecological efficiency, which is the efficiency of energy or net productivity transfer from one trophic level to the next. So the efficiency of transfer is just the ratio of net productivity at one trophic level to the net productivity of the level just beneath. And if the efficiency is the same at each level, it may be close to the same, around 10%, then the productivity at level n plus 1 is equal to the total net primary productivity, the productivity of the autotrophic compartment multiplied by the efficiency taken to the nth power. Okay, and that sort of determines the food chain length. So let's just take a look at it. So we've got the total net primary productivity and the efficiency of energy transfer. Those are two community properties. And the productivity of consumers, and that's the net secondary productivity. Primary means producers, and secondary means consumers. So the total net primary productivity, which is often abbreviated NPP, and the efficiency of energy transfer, that's abbreviated EFF. And this efficiency of energy transfer gets some mean value in a community. That's called the community's ecological efficiency, or you can call it the food chain efficiency. All right, so that efficiency is just the net productivity at one level divided by the net productivity of the level just below it within a community. So we start at the lowest level, the net primary production. That's trophic level 1. That's the net production, not the gross production. And about 10% of that production survives to result in production of the second level, the first order consumers. And that amount is simply the net primary production times the efficiency, which is about 10% on average. And then the third level, the production of the third level, is the net primary production times the efficiency, this right here, times the efficiency again to get the third level. And the fourth is NPP times the efficiency to the third power, taken to the third power to get the production of the fourth level. And the fifth level and so on. So the production at any level, at level n plus 1, for example, is just the net primary production times the efficiency taken to the nth power, because there are n transfers in a trophic chain of n plus 1 length. So here we've got 1, 2, 3, 4, 5. So there are 4 transfers here, 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 and here. So this number is 1 minus the trophic level whose production we are estimating. Okay, so the productivity of n plus 1, level n plus 1, is equal to the productivity of the autotrophs times e to the nth power. Productivity of level n is productivity of autotrophs times e to the n minus 1 power. So you can write it either way, whatever is the simplest to work with. So ecological efficiencies in the marine environment, the efficiencies of energy transfer in different communities, they're somewhat predictable in different types of marine habitats that lead to some broad observations about marine ecosystems. So oceanic ecosystems have ecological efficiency of about 10%. That's this right here. Coastal ecosystems that are non-upwelling have an efficiency of about 15%, and upwelling ecosystems have ecological efficiencies of about 20%. Also, net primary productivities vary somewhat predictably in these habitats. So the oceanic habitat, the net primary productivity is around 75 grams of carbon per meter squared per year. Coastal is about 300, quite a bit higher. Upwelling is a bit higher than that, about 500. So if you do the math with the above equation that we just calculated, you find that the total fish production per unit area in upwelling areas is about 45 times the production in coastal areas, and is about 50,000 times the production per unit area in oceanic systems. And if you multiply out by the total ocean area, these numbers here on the top, you find that even though upwelling area is only 1% of the total area of the world oceans, their total fish production is about 5 times the fish production of coastal non-upwelling areas, and almost 700 times the total production of oceanic systems. So this shows again that the properties of the community, for example, ecological efficiency, can have an enormous effect on the environment and the evolutionary potential of populations within that environment, that community. So if we take E as the ecological efficiency, and then we can repeat our rules above, second order consumer has to forage across 1 over E times the area as a herbivore. Third order consumer must forage across 1 over E squared times the area as herbivore. Fourth order consumer must forage across 1 over E cubed times the area as herbivore. And in upwelling regions, remember E is 0.2, 20%. And we have salmon and baleen whales right at the third trophic level there. Open ocean with 6 trophic levels has an E of 0.1, which means that by the time you get up to the 6 trophic levels, there's very little energy left to be consumed at that level. Continental shelves have an E of a bit higher than that, okay, with 4 trophic levels. Okay, so here we have oceanic, coastal, and upwelling communities. We can add to that. We've got open ocean, continental shelf, estuary, algal coral beds, upwelling, mangrove, and salt marsh communities. You can see the area percent here. of those communities, and this is on a log scale here. So open ocean is close to 100%. And this is productivity, grams of carbon per square meter per year. So open ocean has the lowest. And the highest is mangrove. We've got here algal coral reefs and estuaries. And then we also have salt marsh upwelling areas. And there's continental shelf is quite a bit lower. All right, so this is the general picture of community productivity, energy flow, and production in marine ecosystems. All right, so next we're going to ask why. Okay, why is the productivity different in grams of carbon squared per meter per year? And this productivity here is green productivity, so this is net primary productivity. So why these differences? What determines net primary productivity and what determines the efficiency and net secondary productivity? Net secondary productivity is animals, NSP. And what determines the ecological efficiency of transfer between trophic levels? So that determines the net secondary productivity. At the autotroph compartment here, the efficiency of transfer of light energy into net productivity is due to two efficiencies. Okay, that's the photosynthetic efficiency, which is equal to the gross primary productivity, or fixed carbon, divided by the total incident light, and this efficiency is usually around 1 to 2%. And we also have the net production efficiency, which is the net primary productivity, NPP, divided by GPP, which is the gross primary productivity, the amount fixed. And this is usually around 30 to 85%, dependent on the amount of respiration, which is dependent on the ambient temperature. The higher the temperature, the higher the respiration rate. Why do different species have different photosynthetic production efficiencies? Why do different communities have different efficiencies? Well, part of the reason is adaptive evolution. How species manage their photosynthetic machinery and what turns out to be the most optimal way to invest limiting resources into photosynthesis versus other components of fitness. Another part of the reason is the physical conditions at the geographic location of the community. Okay, so other reasons, basically, we don't know. It's an active area of research. So we can start to understand photosynthetic efficiencies by looking at the saturation curve of the amount of carbon fixed or assimilated. This is also called an assimilation curve, same as fixed carbon curve, which is the grams of carbon per leaf surface area per minute against the incident light intensity, photosynthetically active radiation, from no light down here to full sunlight without clouds. So this is a saturation curve, and the maximum assimilation rate being reached at some light intensity characteristic of the species. So some species reach their maximum at high light intensity, others at intermediate light intensity, depending on how much chlorophyll and photosynthetic enzymes they have per leaf biomass, which depends on how the plants are adapted to the light regime, with different optima in the tropics versus temperate regions, in arid and cloudless environments versus wetter and cloudier environments. So the light optimum for a plant, also depends on the community it occupies and whether other plants are in the community and the other plants in the community. So some species evolve to specialize on low light conditions. On the forest understory or shrub layer or the understory of kelp beds, and have a photosynthetic maximum at lower light intensity. Others specialize on competition for highlight conditions at the top of the forest canopy and others in between. So the light optimum of a plant depends in part on competition for light, on competitive exclusion, and how plants in the community have evolved to partition the light resource and avoid competitive exclusion. So we might expect to see character displacement in light optimum allopatric versus sympatric species of plants. Nobody has looked for this yet that I know of, so it's an area of active research. All right, as a general rule of thumb, plants are capable of converting about 1 to 2% of light energy into carbon assimilation at low to intermediate light levels. But after light levels pass the optimum, that percentage goes down because the plants no longer can use that additional light energy for assimilation. So this is much less than 1% being processed here. This could be like 0.5% if you have high light intensity. So after peak assimilation is reached, no more light energy is able to be used for additional assimilation. So all this light energy here is basically wasted. It's not used. And we also need to take into account the respiration rate. And this chemical reaction is the reverse of photosynthesis. So when net assimilation rate is 0, this means that the gross assimilation equals the gross respiration rate. This is the gross assimilation here, and this is the gross respiration rate. So the net is the gross minus the gross assimilation minus the gross respiration. That's the net primary productivity. Okay, so at this point, this is called the light compensation point. This is the light intensity at which carbon gain just balances carbon loss. That's the light compensation point. Or another way of putting this, net primary productivity efficiency is 0 at this point. There's 0 net production. In order to have positive efficiency, we need more light. So increasing light increases the net primary production efficiency. As we have a higher proportion of the gross carbon gained and preserved as a net gain against respiration, if the respiration rate is constant and doesn't change with light intensity, which is usually the case. So net production efficiency of a plant will increase as light increases. This is the NPP. Light increases until it reaches a maximum at the maximum assimilation rate. At this maximum, light is no longer limiting assimilation, rather some other factors limiting, probably water, or in the marine environment, it's going to be nutrients, so nitrogen or phosphorus or iron or silicate. One broad pattern regarding net primary production efficiency is its relation with temperature. Here on the x-axis. Higher temperatures are associated with higher respiration rates. Respiration rate goes up as the temperature goes up. So if we plot respiration rate and gross primary production against temperature, we get roughly a linear curve for respiration rate and a convex curve for gross primary production with a peak at about 21 degrees Celsius. So this means that the temperature optimum for photosynthesis for many temperate and tropical plants is around room temperature. So the difference between the two curves is the net primary productivity. So the NPP divided by the GPP is the net production efficiency. So general rule of thumb, higher temperatures in tropical communities result in lower net production efficiency because temperature and respiration rates are higher, and so more carbon is lost to respiration. So this curve right here, this line, this red line, represents the carbon lost per respiration. So the higher the temperature, higher the respiration, the lower the net production above the optimum, and the lower the net production efficiency. So the tropics have around a 40 to 60% net primary production efficiency. The temperate zone, closer to 75 to 85% NPP efficiency. Less carbon is lost from respiration as there is less respiration in temperate zones. Temperatures are lower, and this is true for both marine and terrestrial habitats. Also, a general rule of thumb, a higher supply of limiting plant nutrients, higher supply rate of limiting plant nutrients results in higher NPP at all light intensities, but especially at high light intensity, because here nutrients are limiting rather than light, which is abundant. Okay, so different communities have different nutrient supplies, and the nutrient supply rate is a community property, supply being provided by the detrital food web, as we'll see in the next week's lecture. So this results in predictable differences among the major oceanic habitats in NPP and NPP efficiency. In the open ocean, nutrients rain, just drop due to gravity to the sea bottom. Gravity causes nutrients to sink down at the bottom where they accumulate indefinitely. It can accumulate for centuries. So the surface waters in the euphotic zone of the ocean have very low nutrient supply rates in the open ocean, with resulting low NPP values. In coastal upwelling areas, in shallow continental shelf regions, upwelling due to the Coriolis effect, coupled with wind-generated surface currents, this occurs and brings nutrients that have sunk to the shallow continental shelf back up to the surface. So upwelling results in higher nutrient concentrations, lower oxygen, lower temperatures in the euphotic zone, brings colder water up to the surface. And all those result in high NPP of the phytoplankton and multicellular algae, kelp beds, macrophytes, and the seagrasses. Coastal upwelling occurs in eastern boundary currents in 5 main regions. So we're talking about the California current, the Peru-Humboldt current, the Canary current, Northwest Africa, also in the Indian Ocean, due to the monsoon regime, down here the Namibia current. So coastal upwelling can occur year-round, creating high productivity in all seasons, or it can be seasonal, just during the summer. So it's a year-round phenomenon close to subtropical latitudes, because these latitudes are the regions of the subtropical high pressure zones, which create the prevailing winds that cause the upwelling currents. And there are prevailing winds all year round if you're in the subtropics. Outside these subtropical regions, into the temperate zone, those winds from the subtropical highs are strongest during the summer, when the pressure system moves poleward with the solar equator, making upwelling primarily a summer phenomenon in temperate latitudes. So down here, the southern portion, this is year-round upwelling, and up here, the northern portion, it's just summer upwelling, and the mirror image in the southern hemisphere. So because nutrients are abundant, at least during the summer, when sunlight is also abundant, upwelling zones are very highly productive with high NPP and high NPP efficiency, as we saw above in the comparison number in that table. All right, in polar regions, NPP is limited primarily by light, and NPP is very low most of the year, but spikes with very high numbers in the spring when high light and high nutrients coincide. Some polar regions have high upwelling year-round, so for example, the North Atlantic deep water upwells in the Antarctic all year round. And so they're not limited by nutrients most of the year, rather by light. Therefore, there's a very high productivity in the euphotic zone during the spring and summer when sunlight is abundant. The rest of the year, productivity is quite low. So outside upwelling zones, there are some predictable global patterns. Okay. In the tropics, sunlight is always abundant and never limiting, penetrating deeper into the water and maintaining a deep euphotic zone, the tropics. Rather, nutrients are limiting year round. And productivity is steady but low all year round. That's in the tropics. Okay, the main reason tropical nutrient abundance and recycling are low, sunlight heats the upper layer of water, creating a thermocline that stabilizes most of the euphotic zone, okay, and prevents mixing with deeper cold water, and that prevents nutrient return to the surface, lighted waters. So nutrients generally sink by gravity out of the euphotic zone. In the stable thermocline of the tropics, that prevents mixing that is necessary for their return. There are exceptions to this low productivity pattern in the tropics, and these are in equatorial upwelling areas caused by the trade winds and coastal upwelling, but this generally is in the subtropics and temperate zone, not in the tropics. And 3, coral reefs. Corals have symbiotic algae that contribute to productivity and prevent loss of nutrients from the system, so efficient nutrient cycling maintains very high NPP in tropical reefs. In the temperate zone, outside upwelling areas, which is the vast majority of the temperate zone, the pattern goes like this. So during the winter, nutrients are abundant in the euphotic zone. Okay, that's this purple curve, nutrients. They're abundant in the euphotic zone. So this is the euphotic zone. Okay, so in the winter, nutrients are abundant because the sun is at a low angle. The surface is cold. There's no thermocline, and there's plenty of mixing. So the nutrients from deeper areas come up to the surface and mix because of the lack of the thermocline during the winter. But productivity is basically nil down here because the low angle of the sun and light penetration is low. The light compensation depths is very shallow, so the euphotic zone is very thin in the winter. So very thin euphotic zone in the winter. Not much productivity. During the spring, we have a bloom, spring blue of phytoplankton. The sun's angle is more direct, the compensation depth is greater, so you have to go deeper before you run out of light, and the euphotic zone is thicker and there's more incident light, and nutrients are abundant still. So NPP peaks right in the spring because you've got abundant light and abundant nutrients. Deeper euphotic zone, I've got phytoplankton, zooplankton, and nutrient-rich water in the spring. And a thermocline starts to develop. By the summer, this high net primary productivity of phytoplankton blooms depletes nutrients in the surface euphotic zone. And so NPP now drops to low levels because the nutrients are now limiting. Even though there's a lot of sunlight, now nutrients become limiting. So nutrients are not able to be recycled from depth because of the high temperature at the surface, which creates a strong thermocline. Thermocline basically means there's a layer of low density warm water floating on top that doesn't mix because it's warm and floats on top. Stays on top because of its temperature, and so there's very little mixing with the colder and nutrient-rich water that's underneath. So here we have the summer, and this purple water is nutrient-poor water now, so there's not much Not much phytoplankton or zooplankton, and there's a strong thermocline, so which means just basically a layer of warm water at the surface, just so warm it just stays there, and there's just no mixing with the nutrient water below, nutrient-rich water below. Then in the fall, there's another bloom, a smaller bloom. The sun's angle is lower, which cools off the surface, breaks up the thermocline, and allows some mixing, bringing nutrient-rich waters back to the surface. The compensation depth is a little shallower, but not as shallow as in the winter, so there is another smaller burst of productivity in the fall. Still some sunlight, and the nutrients are much more abundant because the thermocline breaks down and there's more mixing. And then we return to the winter conditions. Sunlight goes down. We've got no thermocline at all. There's plenty of mixing, but there's no photosynthesis, so those nutrients remain at the surface in abundance. So here we have fall. We've got a smaller bloom of phytoplankton and then zooplankton in the fall, because the thermocline breaks down a bit, so there's more mixing and more nutrients come up to the surface. So we have more nutrient-rich water at the surface in the fall. So in temperate zones without upwelling, most NPP occurs during the spring transition, during a burst in April and May, usually with a second smaller burst in the fall. So bloom here and in the fall. That's outside the upwelling zone. In temperate zones with upwelling, this high NPP occurs all summer long. All summer long. No depletion of nutrients, and in the subtropics, the high productivity can occur all year around if the winds from the subtropical high are strong all year, as they are in these regions, for example, the California current off California. So because of upwelling, this high productivity occurs all summer long at least, and then in some areas, more equatorward areas of the eastern boundary currents, occurs all year around, and that's the reason for the higher NPP efficiency and higher NPP in upwelling regions. We've got a combination of high nutrients and high sunlight together all summer long. All right, so those are the basic patterns with NPP. What about secondary productivity? That's the animal production. That's production of biomass by the heterotroph component of the community, the animals. Okay, so NSP, this net secondary productivity, is ingestion minus egestion minus respiration. That's this net secondary productivity. So there are four efficiencies in the heterotroph compartment. These are the consumption efficiency, ingestion divided by the total NP of the previous trophic level, the assimilation efficiency, assimilation is ingestion minus egestion. Net production efficiency is net production NP or NSP divided by assimilation. Then the overall efficiency is the product of these three efficiencies. That's NP divided by ingestion. This is called the gross production efficiency, which is probably a confusing name since the numerator is net production, not gross production. So there's some patterns known regarding some of these efficiencies. Okay, so these are just a basic terminology, so you can just look through those and see what the calculations are. It's just very simple, just multiplying the efficiencies together and then just seeing that the units cancel out. So gross production efficiency ends up being the net production divided by the ingestion. That's the gross production efficiency in the heterotroph compartment. And again, the ecological efficiency of the trophic level n+1 is the net production of ns1, n+1, divided by the net production of the level just below it. So you multiply together and you see that these units cancel out. Okay, so here are patterns of assimilation efficiencies. Okay, the ability, which is basically the ability to digest these food items. Okay, so vertebrate eaters, they have about a 90% assimilation efficiency. They do not digest bones or hair or nails, etc., but they digest all the rest, which is about 90%. Consumers of crustaceans or insects have about 70 to 80%, a little bit lower assimilation efficiency. They do not digest the chitin of the exoskeleton of arthropods. Grazers and browsers of seagrasses or terrestrial wetland plants have 60 to 70%, a bit lower, assimilation efficiency of young vegetation, but only 30 to 40% of older vegetation that contains more indigestible material, such as woody lignin, which is indigestible. Not too many wood eaters in the ocean. So net production efficiency is primarily determined by the type of temperature regulation in an animal. This is determined by the respiration rate, okay? Different animals have different respiration rates in order to regulate their temperature. So there's a high respiration rate in endotherms, which regulate temperature by respiratory metabolism, which maintains body temperature. So high respiration rate generally results in a very low net production efficiency. Most food is used to maintain body temperature rather than for growth or reproduction. So assimilation efficiency can be very low, 1 to 6%, or essentially zero for adult males of many species. Females who invest in eggs or gestation, of course, have a positive net production efficiency, but it's still low because respiration rate must be high to maintain body temperature, especially in temperate or polar climates. So poikilotherms, or cold-blooded animals, they have a much higher net production efficiency, way higher than endotherms, since they don't use respiration to maintain body temperature. So here are two properties of communities that we've discussed today: net primary productivity and ecological efficiency of energy transfer. So these are properties that arise from the functional traits of the component species in the community, traits that arise from species surviving and reproducing in the struggle for existence. And these community properties also create limits and opportunities for the species that compose the community, like, for example, predator consumption rates. Okay, well, I think it's time to wrap things up for today. Next week, we're going to continue with at the community level and within ecosystem ecology. So we've talked about energy flow this week. So next week, we're going to talk about material flow, nutrient cycling through ecosystems. And it will be motivated by discussion of the Cambrian explosion, which is the most important evolutionary event on planet Earth and what has created the planet and the living things on the planet as we know them today. So we'll see you next time.